Out of all the groups of species on Earth, few are more important to humans than pollinating insects. Pollination is worth around $220 billion to the global economy each year. That's about the GDP of Greece, and many of the foods that we take for granted would not be available without it. However, for a number of years, pollinators have been under threat. The honeybees that pollinate most plants have been suffering from a combination of disease, pesticides, and parasites, while the other pollinating insects have not adapted well to modern farming practices. When faced with a complex community that's under threat, conservation biologists have to understand how those threats act. This means measuring how the number and diversity of species in the community changes in relation to threats across different geographical locations. Let's take the example of pollinators. There are at least 1,500 species of pollinators in the UK alone, and they're not easy to count even when you're working on only one species. However, scientists have found some methods that can at least provide an idea of what sort of pollinators are found in different places. The first method is the focal floral observation, or flower insect timed count. This simply involves sitting and watching pollinators as they come to visit certain species of plants. By using the same size and same species of plant in all cases, these observations can give a measure of how many individuals from each species are visiting flowers. Another method is to use what we call pan traps. These are small bowls that are painted bright colours to resemble flowers and then filled with water and a small amount of detergent, like washing up liquid. Pollinators mistake these for flowers, they fly into the water, and then they can be collected at the end of the day. One way to make better use of these pan trap samples is not to simply identify the species by eye, which can be almost impossible when many species are almost identical, but to sequence their genes. There are some genes that are common to almost all animals, and this technique looks for small differences in those genes that act like a barcode to identify different species. Analysing large numbers of organisms, such as those collected from a pan trap, is called meta-barcoding. This method produces a comprehensive list of the species that we've found. Other new techniques being developed here at the University of Leeds include ways to listen out for passing insects using microphones that record sounds in the environment. And this technique is called bioacoustics. Bioacoustics listens out for the sounds that insects make as they fly. Our research group uses a device called a solo recorder, built based on Raspberry Pi computers to record over the course of a day. Here you can see our team deploying one of our bioacoustic pan traps. Rather than the insects drowning when they visit our brightly coloured plate, their sound is recorded and then they are free to fly away. The result is thousands of hours of recordings that only require us to visit the site twice, once to set up and once to take down the equipment. But it's then that the harder work begins, because all of those hours of data have to be analysed. Now, rather than listening to all those sound files ourselves, we're training computers to recognise the buzzes of insects using a technique called machine learning. We can show a computer algorithm an example of what we're looking for, and then give it a much larger data set to hunt through to find more. As you can tell, there are many ways to count bees. Some are expensive but efficient, and others are cheap but labour intensive. But all provide valuable insights into how pollinator communities are responding to a changing world.